Okay, so I think we'll go ahead and start now. So welcome everybody to the Biophysical Techniques lecture series, and we are at talk number 4, which is me, Jon Howe. I work within the Light Microscopy Facility at the LMB, and I will be talking about SuperResolution Microscopy. So before I get into the techniques, I thought it was useful to discuss the length scales in biological imaging techniques. So depending upon your sample of interest, obviously you would use different imaging techniques. So if you were looking at whole tissues or organisms, then you would use medical imaging techniques such as, actually I'll get my pointer, there we go, there we go. So you would use medical imaging techniques such as PET scanning, MRI, ultrasound. If you're interested in small protein or small molecule sort of resolution, then most people would opt for electron microscopy because that gives you the highest resolutions. And if you're sort of in the middle, that's where fluorescence microscopy comes in. So typical fluorescence microscopy techniques you will have heard of things like wide field microscopy, confocal microscopy, and these techniques are great, and they have the advantage, although their resolution is not as good as EM, they have the advantage that they are compatible with live cell imaging, and there are also multiple colors of photon, so you can multiplex, you can look at multiple targets at one time. The problem with light microscopy is the optical resolution limit or diffraction limit. This is just a hard limit of the achievable resolution, and in order to overcome this, you have to use clever techniques which are collectively called SuperResolution microscopy, and I'm going to discuss some of the principles of these techniques in today's talk. So what is this optical resolution limit and why does it exist? So as I mentioned, the optical resolution limit is also called the diffraction limit. So that gives you a hint as to the reason why it exists. So because of the wave nature of light and the limiting apertures in the imaging system, when you take an image of a point source of light, which itself would be around a nanometer in size, the image itself is blurred out. And this blur is called the point spread function. Now, it's considerably larger, like several hundred times bigger. This is the what the PSF looks like in the XY and also in an XZ profile. And the first thing to point out here is that the particular lookup table that I'm using emphasizes the dimmer things, the outer elements, so that you can actually see them. But what we're most interested in is this central peak, this central maxima, which, as you can see, is taller in the Z direction than it is broad in XY. And this is because microscopes have poorer Z resolution than XY resolution in general. So the width of this central maxima directly influences your resolving capability. And what you have to imagine is every point source in your object is substituted by a PSF in the image. And the closer these PSFs come together, they will reach a point at which they are no longer resolved. And there are various resolution criteria which are used, and I've listed a few here: Rayleigh, Abbe, Sparrow, full width half max. It's kind of arbitrary which one you choose to use. In this talk, I will mostly be using the Abbe resolution equation. And I will also talk about the full width half max, which is the width of the central maxima at half maximal intensity. And the Abbe resolution limit and the full width half max come out to be similar values. And this is where, sorry, I click forward and my slides don't progress when I've got the pointer, which I haven't used before. So I have to keep on hitting my space bar, so I keep on forgetting. So the lateral resolution, when you plug it through these equations, comes out between 200 and 250 nanometers. Axially, it's bigger, typically 2 to 3 times bigger. And that's in the best case scenario with the highest quality lens, should we say. So if we look at the lateral resolution equation, or the Abbe resolution equation, this is it. This is Ernst Abbe. This is his memorial in Jena, in Germany, and this is his resolution equation. And if we blow it up, you can see that the resolution, or D, is equal to the wavelength of light divided by 2 times n sine alpha. Now this term n sine alpha, which is the refractive index times by the sine of the half angle of collection, is also known as the numerical aperture. So if you're interested in the numerical aperture, which is the light gathering ability of the lens, then you could look at Nick's talk earlier on in the series where he describes this. But essentially, if we substitute this NA into this equation, we end up with lambda over 2NA. And if we put some numbers into this, so typically in light microscopy, the shortest wavelengths that you would use are in the 400 to 500 nanometer range. And the NA is often in the order of 1. Sometimes it's a bit less, sometimes it's a bit more, typically around 1. And if you put those two values into this equation, you end up with the lateral resolution of around 200 to 250 nanometers. And if you wish to exceed that, you need to do SuperResolution Microscopy, or EM. So SuperResolution Microscopy was essentially deemed important enough to award these 3 guys the Nobel Prize in chemistry in 2014 for their contribution towards the field. And there are many super-resolution techniques. And what I've done here is try to break them down into the 3 core techniques or categories. And exactly, these are the sort of initial or original super-resolution techniques. And there are many, many more techniques now, some of which fall within one of these categories, but work slightly differently. Some of them fall within multiple categories, but I'll go through each of them in turn. So we have structured illumination microscopy, or SIM, stimulated emission depletion, STED microscopy, and localization microscopy, which I'll sometimes refer to as LM. So if we start with SIM, so SIM microscopy works like this. So in order to understand SIM, you first have to appreciate that your sample is a complex diffraction grating, and the information encoding the finest details structures in your sample is diffracted beyond the collection angle of the lens. So that light is not collected by the lens and therefore cannot contribute to the image formation process. So your lens essentially acts as a frequency filter. So the highest frequency information just cannot be captured by the lens. And what SIM aims to do is to shift this high spatial frequency information to low spatial frequencies so that it can be captured by the lens. And it does this by using patterned illumination. Now I've tried to diagrammatically depict this in the center panel here. So if you imagine pattern 1 is your sample, and this pattern, obviously you can see it now, but the frequency is so high that it's too fine to be resolved by the microscope. If you illuminate your sample with pattern 2, which is the finest pattern you can project into your sample, so it's right at the resolution limit, then these 2 patterns will interfere to generate the image that you record, which is pattern 3. Now, pattern 3, as you can probably see, contains this lower spatial frequency element. You see these stripes, which are going from sort of 8 p.m. to 2 a.m. And these essentially encode the super resolution information. And because we record this SIM image, pattern 3, and we have a knowledge of pattern 2, we can compute what pattern 1 was. But in order to do this, we have to take quite a lot of images. So it's no good just taking one, because you would just end up with a stripy image, and the patterned illumination is inherently orientated in a particular rotational angle. So what we do is take multiple images where we shift the phase of the pattern, and then also multiple images at different rotations as well. And then we take all of that raw data, we put it through computer algorithms, which then compute the super resolution image. And at best, SIM can achieve a 2-fold improvement in resolution. That's both in X, Y, and Z, which gives you a resolution down to around 100 to 130 nanometers in XY, and 250 to 350 nanometers in Z. And because there's a 2-fold improvement, what I've done is modify here the lateral resolution equation to lambda over 4NA, just to demonstrate that, to reflect that. So we're very lucky to have a SIM microscope here at the LMB, which is made by Zeiss. It has 4 lasers for blue, green, red, and far red excitation. It has several high NA lenses. The resolution in SIM is still linked to the NA, so you want to start with as higher NA as possible. The camera is very quick, which is helpful because you have to take a lot of images. It's also very sensitive. And then there are several gratings, which are the mechanical components, which you can select the optimum grating in order to project into your sample the finest possible pattern. In principle, you can do 4 colors. The important thing is just to get 4 really well labeled components. And as I said, the resolution is still limited by NA and still limited by wavelength. So if you're only planning to do a couple of channels, then it will be better to use shorter wavelength rather than longer wavelength channels. So, for example, use blue and green rather than red and far red. Here's an example of a 2-color SIM image. So on the left is the wide field image, on the right is the SIM image. This is a cow artery cell, essentially, which has been labeled with actin in green and mitochondria in red. And you can hopefully clearly see that the image resolution is enhanced in the SIM image compared to the wide field image. I should say now, because I forgot, if you have any questions, please put them in the Q&A section, unless you are a speaker in the series, in which case you will need to use the chat function. Here is another example of SIM imaging. So this is 3-color and 4-color at the bottom. So these images were given to me by Aggie, formerly of the Randall Lab. In the Randall Lab, they're interested in how cells deal with intracellular pathogens and the process of autophagy. So in both of these examples, the bacteria, which in this case is Salmonella, is labeled in blue, and then the other channels correlate with other proteins which are locating to the kind of capsule which is encapsulating the bacteria. So the 4-color example at the bottom is just to demonstrate that it's possible. If we look at the 3-color example at the top, what's interesting here is that in the wide field example, which is to the left, there's a lot of yellow. There's a lot of overlap between the green and the red signal. Whereas in the SIM image, when the resolution is enhanced, the green and the red are resolved from one another in a lot of areas. And this indicates that these 2 proteins, galectin-8 and ATG13A, are locating to slightly different subdomains, which obviously is interesting. And this is exactly sitting in the sweet spot where a twofold improvement in resolution actually reveals something new. Okay, so next let's talk about STED. STED is quite cool because STED microscopy gives you a super resolution image straight away, because what you actually do is engineer the point spread function to make it smaller. This is how STED microscopy works. So a STED microscope is essentially it's kind of like a fancy confocal. So in a regular confocal, you would have an excitation beam, which is focused by your objective lens down to a small spot in the sample. And then that spot is raster scanned over the sample using some scan mirrors. And then the photo multiply tubes or whatever detectors you're using record the signal, and that signal is attributed to the location where the scan mirrors are pointing in order to build up the image. And normally your excitation spot would generate an emission spot, which is more or less the same size, slightly longer, slightly larger because of the longer wavelength. But instead, we obviously want to somehow make that emission spot smaller. And we do this by using the STED laser. So in the microscope, you have an additional laser, which is on a longer wavelength, which is coupled into the excitation beam. So they're aligned on top of one another, essentially. But the STED beam is kind of modulated such that at the plane of focus, it doesn't form a regular diffraction limited Gaussian profile spot. Instead, it forms this donut shape. And this donut shape is helpful because it enhances the resolution. And it does that by inducing the process of stimulated emission. So the easiest way to explain stimulated emission is to look at this electronic state diagram on the bottom left. So if you excite a fluorophore, it goes from the ground to the excited state, and then ordinarily it would drop down to the ground state and emit a photon. And the exact energy drop defines the wavelength or the energy of the photon which is emitted. And because there's various different vibrational levels, you generate a spectrum of emission. If you bombard the excited state photon with high intensity laser, you can stimulate the sample to emit a photon of the same wavelength of that laser. And this is the process of stimulated emission. So what this means is that all of the fluorophores beneath this high intensity STED laser are induced to undergo the stimulated emission process. Whereas the molecules in the middle, where there's zero intensity in the STED laser, those molecules can undergo the regular fluorescence process. But they're obviously in a more restricted area. So that means your effective emission spot becomes smaller. So practically what we do is we select a STED wavelength at the tail end of the emission spectrum of the dye, and then we detect only the shorter wavelength emission coming from the dye, which is the fluorescence emission. And I should say stimulated emission, in case you're interested, is what the S and the E in the laser acronym stands for. The cool thing about STED is that you can tune the resolution. And you can do this by adjusting the STED laser power. So the STED laser power is denoted I here. Essentially, as you increase the STED laser power, the size of that emitting volume gets smaller and smaller and smaller, so your resolution gets better and better and better. Now, we can also modify the Abbe equation to include this additional term here in red, which contains the STED intensity, but also contains this value here, which is the saturation intensity. This term, saturation intensity, refers to the STED power required to quench emission by 50%, and it's specific for the fluorophore. So each fluorophore will have a saturation intensity. And essentially, the easier it is to saturate the fluorophore, the easier you can improve the resolution. And typically, instead, you can achieve resolutions in the sort of 25 to 80 nanometer range. And in this particular way of working, only the XY resolution is improved. The Z resolution remains the same as it would do on a confocal, although there are ways of improving the Z resolution, which I'll come on to in a second. I have mentioned here that you can get resolutions in down to as low as 10 nanometers by STED, but for biological samples, that's often not feasible because of the labeling methods used, which I'll come on to later. So as I mentioned, the STED is basically a fancy confocal, so the image is still built up via point scanning, but this is actually quite relevant because if you consider this location in the sample with the yellow circle, this location, these fluorophores in this location have undergone multiple rounds of excitation and depletion by the time the imaging spot actually reaches it. And the result of this is bleaching. So it's very important that you make your sample as photostable as possible. Another thing which may have already occurred to you is the fact that the imaging spot is much smaller instead, and therefore the number of fluorophores is going to be much less. So your signal is much lower when you turn on the STED laser. And both of these things mean it's very important to make sure your labeling density is really high and your fluorophores are really resistant to photobleaching. Now, the nice thing is we can also improve the Z resolution instead. We do this by splitting the STED laser into two paths. So the STED laser comes in here, it gets split in the 3D and the 2D path. In the lateral path, it's modified to produce that donut at the plane of focus, which is represented here. And in the 3D route, it's modified so that at the plane of focus, it forms these clouds above and below the focal plane. And when you combine these two STED beams again, at this point, you essentially end up with what's sometimes called a 3D donut. So essentially it's a sphere of high STED intensity, which is basically restricting emission to the very center. So the resolution can be improved in both 2D and also in 3D. You can even achieve an isotropic resolution of maybe 125 nanometers. So, excuse me. So we have one of these microscopes at the LMB. It is built around a Leica SPA confocal, but it has some extra things which makes it a STED. Most of those extra things in this image are to the left of the scan head, over here basically. And those additional components include a tunable, pulsed supercontinuum or white light laser, which is used for excitation, several high power STED lasers, and although this isn't strictly speaking always true, but generally speaking, you need one STED laser per imaging channel. So that's, in this case, 592 depletes green dyes, 660 depletes red dyes, and 775 depletes far red dyes. There is the 3D STED alignment unit, which is this box to the left, immediately to the left of the scan head, which essentially puts the modifies the incoming STED light so that it forms these unusual shapes, and also aligns them on top of the excitation beam. There's also gated detection in the scan head, which further enhances the resolution. This is just a confocal though, so scan time scales with frame size. But the important difference here is that if you wish to achieve improved resolution, so super resolution, you need to make your pixels a lot smaller. And if you maintain the same pixel dwell time, this means you're going to scan a lot slower. So the example here is if your pixels are 20 nanometers, which is 4 times smaller in X and Y than 80 nanometer pixels you would typically use to achieve diffraction limited imaging, then your scan time will be 16 times slower. Now, there is a resonance scanner on the system, but this resonance scanner results in very short pixel dwell time. So it's only really relevant if you have a very, very intense signal. So here's some microtubules, the first of many microtubules, which I'll be showing you. I've labeled these microtubules, which are in Hela cells, with an antibody against alpha tubulin, and then a secondary antibody, which is in the green part of the spectrum. And then you can see the confocal image on the left and the STED image in the middle, and the merge on the right. So, I mean, hopefully it's clear to see here, even over Zoom, that the resolution is significantly improved. If I blow it up, you can see up here what we might have concluded to be 2 microtubules is actually 5. There's a pair just here, and then a triplet of microtubules here. And what we'd quite like to do is to estimate the resolution that we're achieving, and we can do that by taking a line profile of the microtubule and seeing how thin it is. So if we do that on this microtubule down here, then what you see in the confocal image, the full width half max is 250 nanometers. This is essentially showing you what the width of the PSF is. And that value is entirely expected for confocal in the green wavelengths for this particular NA of lens, which is around a 1.4 NA lens. But in the STED image, the PSF is considerably narrower because the resolution has improved, and it's down to 90 nanometers. Now, 90 nanometers is good, but the actual diameter of a microtubule is 25 nanometers. And the reason for this discrepancy I will come on to towards the end of my talk. To demonstrate that you can do multicolor STED, I've got an example here from Jake, formerly of the Gregor lab. Excuse me. This is an image of rat hippocampal neurons, which have been labeled with 2 proteins, or 2 antibodies directed against 2 proteins, both of which locate to the postsynapse. And what you can see, hopefully more clearly in the blow-up, is that in the confocal image, there's a lot of overlap between these two proteins. So in green, we have the GLUA1 receptor, and in red, we have this thing called PSD95. And you can see here we have a lot of yellow co-localization. But when we turn on the STED, a lot of that yellow overlap goes away. And in fact, we see much more resolution, particularly in the green channel. It's quite dotty, and the green protein sort of decorates around the edge of the red protein. Here's another example, also from Jake, similar culture, but now the PSD95, the postsynapse marker, has been labeled in green, and the other protein here is bassoon, which is a presynaptic marker. So in both of these examples, we're actually looking down the synaptic cleft. So what you can see is obviously in the STED image, the resolution's improved substantially. The level of yellow signal is basically dropped away to 0. And what we're looking here is in the cleft where the neurotransmitter will be diffusing across. And finally, here's a 3-color example from Sophie from the CIMR. This sample has been labeled with 3 antibodies directed against receptors which are being trafficked through recycling endosomes. I don't know specifically what the receptors are, but what you can see if I blow it up is that the resolution significantly improves when we turn on the STED here. So whereas most of these objects just look like blobs, when you turn on the STED, you can start to see rings and other various structures. And also the degree of co-localization changes because your resolution is enhanced. And actually this highlights an important thing, which is when you go to super resolution, your resolution is improved, but it's important to do control experiments to ensure that the chromatic aberration in the system is not imposing a mislocalization of the proteins. So this has been done on this data set, so it's already been corrected. So what Sophie would have done would be to label one of these proteins with 3 secondary antibodies in 3 different colors, so that she could take an image, a STED image, and characterize the chromatic aberration, so that she could then apply a correction to a triple labeled sample to ensure that the enhanced resolution could be made best use of, essentially. So the final technique I'm going to talk about today is localization microscopy, which is actually an array of different techniques. Even when it was first invented, there were 3. I am going to focus today on Palm and Storm. But the overriding principles of localization microscopy are the same, regardless of whatever specific technique you're using. And this is the principle. So if you have a number of fluorophores which are unresolved from one another because they're too close, what we would really like to do is image them one by one. And that's what you do in localization microscopy. You contrive somehow to have the situation whereby most of the molecules are switched off at any one time, and you acquire a time series where in each time series just a small subset of the molecules are switched on, and those individual molecules are monodispersed, so they're not overlapping with one another. And this enables us to locate or localize the molecules by computationally fitting this spot to determine a centroid position. And if you do this for every frame of your movie, and I should say the movie it will typically be tens of thousands of time points long, then you eventually have a lot of centroid positions which can all be superimposed into one super resolution image of enhanced resolution. And the achievable resolution here is again in the sort of, you could get a localization precision in the 10 nanometer range. Typically your image resolution will be in the 25 to 40 nanometer range. The Z resolution can also be improved, and I'll show you one technique which we have used in the lab to improve the Z resolution. So what defines your localization precision? Well, the answer essentially is photons. So the more photons, the better. So this equation here shows you the localization precision, and it contains 3 terms here, and these are uncertainty terms. They're noise factors. The first one is the photon noise, the second one is the effect of pixelation, and the third one is the effect of the fluorescent background. Now, if the microscope is designed appropriately, then the pixelation, the pixels are sufficiently small, so that's not a problem. And the background, if the sample is designed appropriately, the background will be sufficiently low. So these 2 terms essentially become very small, and it's this first term that really dominates. And if we if we simplify the equation, just taking into consideration this first term, we end up with this simplified equation down here on the bottom right corner. So your localization precision is approximately equivalent to the width of the focus spot divided by the square root of the number of photons. This is demonstrated in this in silico data here. So at various background levels, the more photons you detect, the better the localization precision, the smaller this becomes. And this is pretty obvious really when you look at some images. So n here is the number of photons, and you can see if we take an image of this spot, the more photons we collect, so either the more excitation power we put in or the longer the exposure time, the more clear it is where the center of the spot might be. So the better our localization precision can be. So in practice, we have an image and it's acquired on a camera. And then we have many images, as I mentioned, many tens of thousands of images. And those images are run through computer processing and Gaussian fitting, which calculates the centroid position of each of the diffraction limited spots. And then that is represented in the higher resolution map, which is the localization microscopy image, or the super resolution image. Now, how you represent that in the LM image is kind of up to you. Sometimes people will just use a cross, so the centroid position. But what's often done is the localization precision is factored into the appearance of the image. So what often is used is a Gaussian blob in the localization microscopy image. So every spot is represented by a blob with a Gaussian profile, and the size of that blob and the intensity is linked to the localization precision. And this ensures that poorly localized molecules don't contribute too much to the appearance of the image. So poorly localized molecules will be represented by a broad, dim spot, and very well localized molecules where many, many photons were collected are represented by a more narrow, bright spot. So as I mentioned, there's a few different super resolution techniques that fall under the category of localization microscopy. One of them is called Photoactivation localization microscopy, and this uses optical highlighter fluorescent proteins. These are fancy fluorescent proteins, derivatives of GFP and M cherry and the like, which you can basically control the photophysical properties of. You can control whether or not they are on or not, or what color they are. So there are two main classes. There's the photoactivatable fluorescent proteins, which are represented to the top here, and then there's the photoconvertible fluorescent proteins, which are on the bottom. So the photoactivatable fluorescent proteins, things like PAGFP, initially start in a dark state. So if you irradiate them with blue light to image them in the green channel, you won't see anything. It'll just be completely dark. But if you trickle in a low intensity of 405 or UV light, you can induce a photoactivation process, which physically changes the structure of the chromophore of the GFP to turn it into a fluorescent version, which is green. And then what you can do is extract as many photons as you can from that activated GFP, and then ultimately it will be bleached. And then what you hope is in the next set of frames, another PAGFP, in the case of this example, will become activated, and then you can image and localize that, and so on for many tens of thousands of frames. The obvious benefit to this is you can multiplex. So you could have, say, PAGFP to label protein 1 and PAM cherry to label protein 2. But the downside is that you cannot tell which cells are expressing your protein. So this isn't a problem if you have a stable cell line, but if you're doing transient transfection, then you have to basically search around in order to find a cell that's even expressing, and find a cell that's expressing at the right level. So if you only need to do single color palm, or single channel, should we say, then it's actually easier to use these photoconvertible fluorescent proteins, which work in a similar way, but they start in a fluorescent state and through activation are converted into a different color. So the examples here are all of green to red converting fluorescent proteins like EOS. So if you imagine you'd have a whole field of view of EOS molecules, which are all visible in the green channel, but if you image in the red channel, you would see nothing. But if you trickle in a little bit of UV light, you will randomly convert a small subset of those EOS molecules from the green to the red version. And then that can be imaged, the centroid found and bleached. And then you move on to the next frame, essentially, where hopefully another molecule somewhere else will be converted to the red form, and then you can image that one. So, although the way in which you achieve the monodispersed state is different, the principle is the same for both photoactivatable and photoconvertible fluorescent proteins. Another technique, another localization technique which is commonly used is called storm or destorm. And this doesn't use fluorescent proteins, it actually uses organic dyes like Alexa 488 or Sci5. And this means that you don't need to do any cloning, you don't need to have a genetic fusion, you just need to have antibodies directed against your protein of interest. So it's often favored for that reason. And the way it works and the property of the dyes that it exploits is the fact that many of these commercially available dyes can be induced to undergo this photo switching process. And this occurs in the presence of millimolar concentrations of reducing thiols like beta-mercaptoethanol. And to explain this, I've got another electronic state diagram on the bottom left and also a movie which I'll show you. So as we've already discussed, the normal process of fluorescence occurs like this. So you have a ground state molecule and it absorbs a photon to the excited state, and then it emits a photon in the way of fluorescence to go back down to the ground state. And this will happen many, many hundreds of times or thousands of times, which is great. But there is a fixed or slim probability that the fluorophore will return to the ground state via this triplet state here on the right. And it happens that this triplet state is sensitive to reduction. And if it's reduced by these thiols, it produces this long-lived off state, so-called radical anion. And the idea of the storm experiment is to push or shelve the majority, like 99.9% of the molecules in this off state. So only a few molecules are in the on state in any one frame of the movie. Obviously, it's important to be able to image all the molecules at some point during the movie. And fortunately, it's long-lived, but the molecules will still return to the fluorescent state via this oxidation process, which also happens to be photoinducible with UV light. The thing to bear in mind is that some dyes, like the carbocyanine dyes, undergo this oxidation process very readily. So it's quite hard to shift them into the off state, or shift enough of them into the off state. So what you can do is add to your buffer, in addition to the reducing thiol, you can add some sort of oxygen scavenging system, like glucose oxidase and catalase, which will mop up all the molecular oxygen to, well, most of the molecular oxygen, just to prevent this, this oxidation process from happening too easily. this movie on the right, which I might need to switch off my pointer in order to play. I took this movie of Alexa 647 labeled secondary antibody just stuck onto glass. And what you'll see is when I move to a new field of view, the molecules are very bright initially. There's lots of them switched on, but then they start to get shelved in the off state, and then only a small subset are switched on in any one frame. And I can modify the density of the blinking events by pumping in more and more 405 or UV light. So it's nice because it's tunable. You can increase the density of the blinks so that they're sufficiently dense that you get lots of localizations in each frame of the movie, but not so dense that the localization precision is affected by the overlap of the molecules. Okay, so we have a microscope capable of this storm technique. It just so happens this microscope also has a turf illuminator. It has 4 lasers, quite high power, just like the SIM. And it has high NA lenses because obviously we want to start with the smallest PSF as possible. Has a sensitive camera, which can also go quite quick. One really useful feature of this microscope is the perfect focus system, which is a hardware autofocus system, which is built into the Nikon stand. And this essentially reduces or eliminates Z drift, which is really helpful because an acquisition can take up to an hour. And if your sample drifts within that time, then that can impair your resolution. So PFS really deals with that problem. Obviously, drift can happen both axially and laterally. So we have a method of dealing with the XY drift, the lateral drift, which is this cross-correlation software correction, which happens after we've acquired the data. And finally, on this system, we have a cylindrical lens between the stand and the camera, which essentially allows us to do some 3D position mapping, which I'll show you in a moment. So I mentioned this is a turf microscope, and some of the images which I'm going to show you will be turf images, so I feel like I should introduce what turf is. So total internal reflection, which is the process through which fiber optic cables work, is a physical phenomena whereby if you incline the angle of the incident laser by basically not firing it straight up the middle of the lens, but rather up the edge of the lens, then rather than the laser passing straight into the sample, like it normally would, it instead bounces off. It's reflected off this interface between the glass and the media of the sample. This only occurs if you exceed this critical angle. So below the critical angle, you would get refraction and the laser would pass through. Above the critical angle, you get reflection. And the important thing here is that at the point of reflection, this evanescent wave is formed. And this evanescent wave decays rapidly in Z. So what this means is the excitation light is restricted to the 100 or so nanometers closest to the cover slip. And this is really helpful because what it ensures is that only molecules close to the cover slip are excited. And anything deeper in the cell doesn't ever see the excitation light and therefore doesn't contribute to the fluorescent background, which, as we've already discussed, is important when you want to localize molecules. So the important thing here, though, is that TURF only works if your protein of interest is in the TURF field. So it really needs to be on the bottom surface of the cell, right up against the cell surface membrane, or maybe a membrane protein. So here is an example of some Hela cells which I labeled with a caveolar antibody against a caveolar protein called cavin-1. And this is the TURF image, which you can see has very, very low background, even though there will be a lot of internalized cabin one. We just don't see that. We only see the stuff at the surface. And the storm image looks like this. And hopefully you can see that the dots are quite significantly smaller. And we can blow them up. And we can just keep on blowing up this image. And eventually what you see is the individual caveoli. And they form this kind of cup shape. It's like C shape. And this is the caveoli sort of pinching off the membrane. And in some examples, we can see an actual vesicle that's already been released from the membrane, but it's still quite close to the membrane, so it's still visible in the TURF field. So that's obviously really good resolution. That scale bar is 100 nanometers, so probably the resolution here is 50 or better. As I said, I like to show images of microtubules. So here is a wide field image taken on our TURF microscope of microtubules. This is the storm image. And if we blow that up, hopefully you can see that the resolution is improved. And if we take a line profile across the microtubules, just like we did in the STED example, you can see that in the wide field image, the full width half max is 350 nanometers, which is expected. It's bigger than it was in the STED because we're using a longer wavelength dye. And in the storm example, the full width half max is much smaller. It's down to 70 nanometers. But again, that's still not 25 nanometers, which is the diameter of the microtubule. I mentioned there is this cylindrical lens, which gives us STED mapping. So essentially the cylindrical lens introduces astigmatism, and this astigmatism is reflected in the shape of the focus spot. So on the plane of focus, let me just get my pointer back on, because I think that's maybe helpful. At the plane of focus, the spot is just round. If the molecule is slightly above the plane of focus, it gets extended and becomes elliptical in the x-axis. If it's below the plane of focus, it becomes extended elliptical in the y-axis. So what this means is when we take a storm data set, not only can we localize the centroid position of each spot, we can characterize the ellipticity of the spot, and that basically encodes its Z position, which enables us to go from a storm image that looks like this to a storm image which looks like this. So now we can see that, for example, the microtubules at the periphery of the cell are lower down than the ones that are around the nucleus, which makes sense because right at the edge of the cell, the cell is much, much thinner, but it's still stuck down on the glass. And near the nucleus, obviously the big size of the nucleus means that you have this sort of fried egg shape in the cell. Okay, so here's an example of some multicolor storm imaging. So this was given to me by Kalina, formerly of the MRC Cancer Unit. She was interested in radiation-induced damage and DNA repair. And I think you can clearly see here that in the diffraction limited image, in the wide field image, these two molecules, RPA and RAD51, are not very well resolved, whereas in the storm image, they're very well resolved. Now, the final couple of slides, I just want to talk about a few other important factors. So we've already discussed that photon number is very important for localization precision in localization microscopy, but also it impacts the signal to noise and all super resolution techniques. But there are other things that impact the image resolution, and those include the labeling density and the linkage error. So I'll cover each of those in turn. So labeling density is pretty obvious. So if this smiley face here is your structure of interest and you label it by decorating it with fluorophores, if you don't have very many fluorophores, you cannot resolve the structure. So you need to make sure you pack as many fluorophores on as possible so the image represents the structure with high fidelity. linkage error refers to the distance between the fluorophore and the molecule of interest. And the linkage error is really dependent upon the labeling method you're using. So in the STORM data I presented to you, I was using indirect immunolabeling. So I was using a primary and a secondary antibody. Now an IgG is about 10 nanometers in length. So if I label a microtubule, which is represented here by this red stick, with a primary and then a secondary, it's possible that the fluorophore labeling that microtubule could be as much as 20 nanometers away from the surface of the microtubule, which is obviously quite a long way. And this means that your 25 nanometer diameter microtubule actually becomes 65 nanometers in diameter when you've added on your label. And this is the reason why, regardless of how high how high the resolution was and how high the localization precision was in my STORM imaging, I could not observe the microtubule as a 25 nanometer structure. And that's because it was just bigger by the time I'd labeled it. Now, obviously, there are other labeling options out there. One of the smallest options, obviously, would be to use a fluorescent protein, which has the benefit of being exquisitely specific, but has the downside of the fact that fluorescent proteins are not as bright as organic dyes. So a typical fluorescent protein will give off about 1/10 of the number of photons as an organic dye. And there's also no amplification when you use a fluorescent protein, as there is with antibody labeling. Now, other options would include, say, nanobodies, which are smaller antibodies, which obviously reduce this linkage error. And finally, another good option are the snap tags, halo tags, those sort of labeling methods, which retain the specificity of a genetic fusion, but have the signal intensity of organic fluorophores. And in my final slide, I just want to compare the various methods that I've been talking about today. So we have SIM, STED, and localization microscopy. Moving from left to right, the resolution improves. That's the spatial resolution, but actually the temporal resolution gets a lot worse. And that's just because of how long it takes to acquire the image. In terms of STED, you can actually make that go quite quickly if you're willing to scan a very small area. So it is possible to get the temporal resolution to be quite good instead. In terms of multiplexing, SIM is the most flexible. STED is restricted by the number of laser lines you have, and localization microscopy is restricted by the number of genetic fusions you can make, or the ability to get multiple dyes blinking at the right density in your STORM buffer. In terms of post-processing, both SIM and localization microscopy require post-processing, and therefore you have to watch out for artifacts. Just a few tips here at the bottom before I conclude. So if you're doing super res, you should use a number 1.5 thickness cover slip. This is the thickness of cover slip which most lenses are designed to be used with. And ideally use the low tolerance or high precision versions. If you culturing your cells, obviously, ideally you would get rid of the phenol red because it's dye and it's just going to add to the background. In order to achieve high resolution, you have to have a high refractive index sample. Obviously, this is only true, this is only possible when you're mounting the sample, which is only compatible really with SIM and with STED. When you're doing STORM, you need to leave the sample in media so that you can add on the blinking buffer, as it's called. But if you're mounting the sample, I recommend something like Prolonged Diamond or any high refractive index mounting media with anti-fade agents in it to prevent bleaching. And finally, if you're doing immunostaining, I would recommend increasing the antibody concentration for both the primary and the secondary in order to ensure high labeling density. But obviously, if you're using a lot of antibody, you will need to increase the duration and number of washes to reduce non-specific labeling. So that's it. These are my acknowledgements, which I'll let you read, and I will take a look in the chat to see if I have any questions. I've got a question from Chris. So he is asking, in papers and talks, claims of co-localization should come with a image resolution caveat. They're in this, i.e. they're in the same neighborhood, but may not be interacting directly. Sometimes co-localization is used as an indicator of interaction. So yes, I agree entirely. So two molecules overlapping in the image does not mean they're interacting necessarily. And if images, it would be ideal if everyone would calculate their image resolution and present that alongside the image, because then you could use that as part of the picture when interpreting the image. Obviously, the more and more resolution you get, the more you have to take care of chromatic aberration, as I mentioned, in order to make firm conclusions about co-localization. And ultimately, you know, if you want to detect interaction, there are other techniques which actually kind of have more resolution. Things like FRET only really works if the molecules are within 10 nanometers of one another. Or there's obviously other protein biochemistry techniques which you can use for seeing if things are co-IP, for example. But yeah, that's a good point, Chris. Anyone else? What kind of drift correction method do you use for XY in storm? What is the frequency of the correction? Okay, so we use a cross-correlative drift correction algorithm. So essentially what happens is we have a data set which is maybe 100,000 frames long. And then the software, you create many storm images, many sort of sub-storm images. And because in each of those sub-storm images, you know, created through subsets of contiguous frames, the same structures should be present. If the positioning of those structures has changed, you can essentially correct that by shifting the latter sub-images back onto the position of the initial ones, and then superimpose all of the data to get a final drift corrected image. So that's how the cross correlation drift correction works. So the resolution is, yeah, you need enough localizations within each substorm image essentially in order to accurately and faithfully reproduce the structure, so that you can then reliably identify how much to shift subsequent images by, or subsequent localizations by. I think that's it from the question and answers. I'll just take another look to see if there's anything in the chat. Don't think so. So I think that's everything. If obviously I'm on site for any LMB people, so feel free to drop me an email if you're maybe interested in doing super resolution microscopy, because the earlier you involve me or someone else in the facility, the better, because we can give you advice on how to prepare the sample. We can even provide you with some of the labeling reagents, and then we can obviously train you, and all of those things give us the best chance of success essentially. So yeah, just keep us in the loop if you're interested in doing this, and we'll work together with you. Okay, I think that's it. I don't think we've got any more questions, so you're all welcome to leave if you like.